This photo shows us on March 13th mapping the electric field of the weakly electric fish Nathanemus petersii.
So this is a picture of our mapping apparatus with the computer and electronics on the left, the mapping tank robot and anesthetic in the middle, and the TV displaying an experiment in progress on the right.
There's a camera on the side of the tank to size the distance and the size of the experiment. These structures, along with the 4 small insulated electrodes, minimize the EOD field. Harness them use the indicated along with the inter-electrode distance calibrated in the inset metric and approximately indicated by the color lines.
As you can see in the picture, we are taking the immobilized fish and positioning it for EOD mapping in the tank.
In the top left image, the reference channel EODs are displayed as they were recorded by the analog to digital converter. By comparing the similarities in their displacement, we can come up with a better alignment for each reference channel EOD. On the right is a map of the mirrored electric potential across the given sample size.
So for the result, here is the EOD potential animated in the particular color, and this is the mapping for the EOD.
So this figure here depicts the field measured at all of the points in the experiment. This can be seen more clearly in figure 3, where we focus more closely on the nasal and schnauzen organ regions of the fish. As you can see, the EOD vector extends out, goes in, and loops around and then returns to 0.
Another representation of the electric image of this 1 millimeter sphere as a function of position across the proximal surface of the fish's body.
We compared our simulated electric images with those of Gottwald's figure 1. The image has been partially reproduced at the right. In hindsight, we suspect the measurements they show for the capacitor would be smaller than the M-perturbed EOD were it not for conductive terminals on either ends of the capacitor. We hope to conduct many more comparisons between our data and those of other groups and expand our mapping studies to more regions of this fish and other species.
This project was very important to me because I learned a lot about being on a diverse research team and interdisciplinary science. I learned how entangled science is between disciplines, as well as how to create experiments that question nature with purpose to offer valuable answers to the mysteries of the natural world. And I feel this project helped me in my career goals to bridge my past academic experience in physics with my future in biotechnology and business.
After just a year and a half with a group, I have been exposed to so many different aspects of experimental research, from developing a robotic apparatus to editing programs to caring for and anesthetizing test subjects. Due to this diverse experience, working in an interdisciplinary lab, my research interests and goals for graduate school have evolved, and I am eager to work with researchers from a variety of fields and backgrounds.
I had recently joined the FISH group just a semester. With my background in biology, membrane and action potential are well-known occurrences in cell biology. What is not well-known, however, in biology are how organisms utilize action and membrane potentials to better navigate their environment. This transition is what initially drew me in. Learning how electric fish utilize their discharges was especially interesting to me. Overall, I had a very positive experience with the FISH team and learned a lot.
It's my honor to take part in the experiment with my last semester, where I experienced the different charm of physics experiments than classes. The mixture of the fields also contributes a lot of interesting and attractive experiments, which are waiting for digging by us. Overall, it will be my precious, positive, and happy experience contributing to my physics study life.